Hello,
Hello?
welcome,
Oh, hi.
Will. Am I on to you?
Yes, I can hear you.
Okay. So, Miguel, okay, you have sent me a picture which it says substitution mutation occur in a gene coding for an enzyme. Which of the following choices is most unlikely result? Like, which cannot in this particular situation of substitution mutations. Do you know what is a substitution mutation?
Um, well, what I know so far about a substitution mutation is that a mutation is like exchanges from like one single base to another.
Yes, correct. When the sequence of the amino acid, okay, only
Okay.
one base is changed, then that is substitution mutation. So let's read the question, what it says. Let's read the options. A, the mutation produces a codon that codes for the same amino acids as the original. The enzyme's protein structure is unchanged. Okay, let's read the
Okay.
B. The mutation produces a codon that codes for amino acid with similar chemical properties to the original, resulting in an enzyme with the same function as the original. C is the mutation produces a stop codon in place of a codon for the placement of an amino acid. This produces a protein that is shorter than the original and non functional enzyme. Okay.
Okay.
The mutation produces a codon that codes for amino acid with different chemical property than the original one. The enzyme now catalyzes a different reaction than the original. So basically, D option is the thing that actually happens. Okay, when one of the base is replaced, which cannot happen is option A. Why? Because in option A, they are saying that mutation will produce a codon that codes for the same amino acid. No, it is incorrect.
Okay.
Okay,
Okay.
it is incorrect. So the correct answer for this question is option A.
Okay.
A1 is the correct answer.
Okay.
Okay, Miguel.
Okay.
So
Um,
have you understood the explanation?
well, I submitted it, but it turns out it was incorrect. And then they said that the correct choice is option D.
Okay, so is it the incorrect one?
Option D is the correct answer. Option A was the incorrect answer.
Just a minute. Which of the following choices is most unlikely result? Most unlikely result. Okay.
Um, okay. Okay.
Okay. Actually, I got confused. I'm so sorry. They are asking us that what can actually happen. So D is the option, as I earlier told you, that is actually going to happen. What cannot happen is option A. So yes, the correct answer is option D. I'm so sorry for that.
It's okay.
Okay, so because we are changing one base pair, definitely the codon is now going to change, which is now going to code for another kind of amino acid. It will be different from the previous one. Okay,
Mm, yeah.
so is there anything else that you want to ask?
There's one more like thing to like ask him. I'll show it to you right now.
Okay, so are you sending me the picture?
Here I am.
Okay, okay, I'm waiting for that. Okay. Determine whether the following molecular modification follow the process of transcription or translation. Okay, so whatever changes that we are doing, we need to, categorize them into either translation or transcription. So Do you know what is transcription or translation, Miguel?
Transcription isn't like transcription like or sorry, it's translation. Translation is kind of like saying like a cell that like reads info in RNA.
Okay, so here the transcription is basically the formation of the RNA, okay, from DNA. And then translation is the formation of the protein or the amino acid from the RNA that was formed after transcription. Okay,
Okay.
so they are basically these two processes are in continuation. So let's read the option. Addition of a phosphate group to a polypeptide. Okay, so we need to
Okay.
uh, tell them that whether it's a post transcriptional modification, that is either it's happening after transcription, post means after, or is it happening after translation. Okay, so any guesses?
Well, so like the first one is transcription or translation.
uh, just a minute. They are talking about the phosphorylation process. So it's a post translational modification. That means the correct answer is post translational modification.
Okay. And then the
Okay.
second, it's just because I've already like explained that like I've been kind of like falling behind on this assignment. I kind of need to like I gotta like get some of the answers and I do want to learn, but I need to like get some of these answers before this assignment is like, considered late for the progress report.
Okay. So can we? That's great. Can we move on to the next one?
Oh, sure.
Okay. So they are asking that addition of a poly A tail to an RNA molecule.
Mm.
Okay, so we need to tell that whether it's a post transcriptional modification, that means either it's happening after transcription or it's a post translational modification, like whether it's happening after translation. So is there any guess again for this question?
Oh no, I guess not.
Okay, so here it's the post transcriptional modification, the addition of the poly A tail, that means addition of the adenine groups. Okay, adenines, extra
Okay.
adenines are added. So this happens after the transcription. So it's a post transcriptional modification.
That's for the second one, right?
It's the first one, post transcriptional modification for the
What about the
second option.
second one?
That means addition.
Well, you said the second one is
Okay.
translation.
I'll just repeat myself. Addition of poly A tail to an RNA molecule is post transcriptional modification. It happens after transcription.
In addition of phosphate group to a polypeptide, which one is that one?
It's post translational modifications.
Okay. And now
Post
for the third.
translational.
Okay. Now for the third one.
Third one, cleavage of a amino acid polymers. So hydrolysis, it present a post translational modification again.
Okay. And for the fourth one.
Removal of intron segments from RNA molecule, that's the post transcriptional modification. It happens after transcription.
And then for the final and fifth one.
Fifth one is again post transcriptional modification.
Okay.
Okay, so
Okay.
I think we are done with this question. Now you can submit this one and check for the answers.
Yeah, I got all those correct.
All are correct. So, oh, well done. That's great. Now, is there any other question that you want to ask?
There's one last one, and this might be the most
Oh,
difficult one though.
okay. So let's see if it's the difficult one.
Yeah. They're separated into like a couple pictures, because it's the same question, but I just need to separate it in like different photos since it's a pretty big question.
Okay, okay. So just send me all the things related to that question. We'll just see. Okay?
Right.
All right. Okay. So I just got one picture and then there is second. So just send me all the picture and then I'll begin with the question.
I'm trying to send them all right now.
Okay. So shall we begin with the question?
Yeah,
Okay.
that's all the.
So yes, this looks a little bit difficult because it's a lengthy question. So let's just see that what's this. So it's basically about lac operon. Do you know what is a lac operon? Any idea about it?
A lac operon. Isn't like a lac operon like a pile of genes with one promoter?
Okay. Anything else?
Um, they're like they're the genes in the encode of the operon. They allow like bacteria to use lactose as energy.
Yes, correct. So here what happens that we have separate set of genes. Definitely there is promoter, there is repressor, or we can say like inhibitor, regulatory, structural genes. So they basically helps in the formation of certain enzymes, okay, with the help of energy from lactose. That was correct. Which is actually an inducer also, okay.
Okay.
Lactose is a kind of a substrate for the enzyme, and it also helps in switching on or off the operon. So that's correct. Let's see what they have mentioned. An operon is a cluster of regulated genes in prokaryote. It includes structural, coding for the enzyme, regulatory sites, which are promoter and operator. The lac operon is classic example of gene regulation. So they have basically given all the information. Okay. So let's read what's on the second page. One is RNA polymerase. Two is inducer molecule, that is lactose. Three is promoter. Four is operator. Five is repressor protein. Okay, where is five? Okay. So they have, okay. A lac-operon remains off unless a repressor molecule binds to and removes the inducer from the operator, allowing RNA polymerase to transcribe genes 6, 7, and 8. Okay, so 6, 7, and 8 are the structural genes. Okay, we are going to write very few information about it. So basically, 6, 7, and 8 are the structural genes that will help to manufacture the enzymes that a bacteria requires. Okay, let's read another one. Okay, so we are done with this one. Okay. Miguel, I think what exactly is the question? Where is the question?
Um, well, the question is like saying like the full question is like saying that the operon is like a cluster of regulated genes and prokaryotes, and that includes structural genes, coding for enzymes, regulatory genes, coding for the activator or represents the proteins and regularity sites, promoters and operators. And then there's there here like all bacterial operants, the lac operant is able to produce multiple proteins from one mRNA transcript. The figure below illustrates the lac operant when it is off the top and when it's on bottom.
Okay. There is just one question. Oh, okay. So they are the option. Oh, understood, understood. That they have given the options. Okay, okay. So here, if you see figure A is one such option. We have to choose one option out of these four. So one is RNA polymerase. All right. Two is inducer molecule. Okay. Three is promoter. Three is promoter. Let's see. Okay. Fourth is operator. Five is repressor protein. Okay.
Okay.
Okay. Okay. Just let me just see all the options. Repressor protein, no. RNA polymerase. Two is RNA polymerase. Four is operator. Okay. So just give me a few minutes. I'm just reading, and
Okay. Sure.
one is RNA polymerase. Two repressor protein. Five inducer molecule lactose. Okay. Three is promoter, four is operator, five is inducer, one is RNA polymerase. Okay, so till now, fifth is the inducer molecule lactose, second is repressor, three is promoter, four is operator. Okay, so till now C, let's see the. Okay, so I have read all the option and the correct one according to me is C option. Okay, I'm so sorry this question took a little bit time, but the correct answer should be C.
Don't worry. I understand that. It takes time.
Okay,
Okay. Yes.
so the answer for this question is C option.
Okay. I got it. Yeah, it's correct. I really think.
Correct?
Yes.
Okay, thank you so much. Finally, we are done with all your question. I'm sorry. I'm so sorry if it took a little bit more time. Okay.
That's all right. I understand why, but I appreciate it.
Okay, thank you so much. And Miguel, is there anything that you didn't understand and you want me to repeat it? Is there any kind of a doubt you want to ask?
No, because I took in notes of like what you explained.
Okay, thank you so much. And if you like the explanation, please rate this call. It means a lot to me. And thank you so much. Have a nice day.
And you too.
Thank you. Thank you.